bit about it to give an idea about common hemoglobinopathies. We know that the red blood cell, the major component of this cell is the protein, the hemoglobin, which constitutes more than 70% of the content of the RBC. And this biconcave cell actually has millions of hemoglobin molecules. So we know that this molecule protein is actually 2 alpha chains and 2 beta chains, which are adjoining to each other. And this tetramer is called as the hemoglobin molecule. So the protein structure is such, usually like you'll have the primary structure where you have the amino acids lined up, and then you have the secondary structure where they will fold upon themselves, forming finally the tertiary structure. And 4 of these tertiary structures, which means that 2 alpha chains and the 2 beta chains, when they come together, they are forming the hemoglobin molecule. Now you have the N-terminal from where actually the protein starts, and this is, the nomenclature goes as A1. So basically the first, straight part which is there of the molecule is called as the A, and when you have a joint over here, when it's turning the next turn, at that time it would be like the B will start. And you will have some junctures where you will find something like FG here. You will find that this will be some of the turns which are not acute angle turns, but it's a little close angle turn. So some of the molecules which are very important, like this, I've marked it in red here, the F8 and E7. These are the areas where you will have the protoporphyrin ring, which is attaching itself to the hemoglobin molecule. Now, you will understand that the way this molecule is placed and the protoporphyrin ring is there in the crevice of the hemoglobin molecule, that there will be some amino acids which are going to be absolutely essential. And if there is a change in the amino acid, the mutation will be such that it will not be tolerated, and you could get the production of some unstable hemoglobins. You will have some changes which might be there, which are tolerated, like something like, say, hemoglobin D Punjab, where there is an amino acid change. It is an abnormal molecule, but there is no pathogenic connotation to this abnormal change. So just to remember that you can get various amino acid changes in the hemoglobin molecule. Some are tolerated, some are mildly tolerated, some of them are just not tolerated. And we'll be talking about the globin chain abnormalities here. So when we are actually breathing, that is oxygenation and deoxygenation takes place. When it is deoxygenated, we call it the TOT structure or the T structure. Here, what happens is that you have the alpha and the beta dimers. So there is one junction which is very important, and actually little changes are allowed at the juncture between alpha and the beta dimers. And some of the junctures are such that, here if you get any mutation, then again you can get some abnormal hemoglobins which might give rise to polycythemic variants, or some different variants can be there. So just to give you an idea that some of the mutations changes are tolerated and some of them are not tolerated. Now, this is how the hemoglobin molecule is coordinated. It's a little complex because the hemoglobin molecule has 2 alpha chains and the 2 beta chains, and the production of these globins are not in the same chromosome. So we call it the beta globin locus is actually present in the chromosome 11 short arm, and the alpha globin chains, these are present, the entire locus is present on chromosome 16 at the telomeric end of the short arm. So you can see that these various different types of globins are there. The genes are there basically. So you have the epsilon, then you have the G gamma, you have the A gamma, and there is a pseudo gene over here in between the A gamma and the delta, and then you have the beta. So the way nature is producing these globin chains is such that initially in the embryonic stage, you have the production of the most proximal kind of gene. This gets upregulated. So in the embryonic stage, you will have the epsilon gene, which will be active. And in the fetal stage, you will have the G gamma and the A gamma, which will get activated. And then in the adults, you will have the delta chain, which has a very little production. And then you have the beta chain, which gives rise to the adult hemoglobin. So this is the production of the alpha globin chain here. So you have the embryonic hemoglobins like the zeta 2 and the zeta 1. And then you have some pseudogenes over here, pseudo alpha. And then you have the alpha 2 and alpha 1. So you can see that the alpha which is produced actually has two alpha genes, the alpha 2 and the alpha 1. And the beta actually has one. So which means that at every time, because the requirement of the beta globin versus the alpha globins are, 1:1 or stoichiometrically arranged. So always the production of alpha chains is in excess to that of beta chain. So this is just an important concept which is there. And this is something you need to keep in mind. Now, during the embryonic stage, you find that the hemoglobins which are produced are actually the GOVER1 to start with, which has two zeta chains and two epsilon chains. Then you have the Portland, which has two zeta chains and two gamma chains. And then you have GOVER2, which has two alpha chains. So you can see that when the GOVER is getting formed, you have the alpha chains which are getting produced. So if you look at this diagram, you will find that the alpha chain production starts on quite early and it is kind of sustained. Whereas the gamma chain production in the fetus is high, and after birth only you will find that the reduction of the gamma chain happens and the beta chain actually takes over just when the child starts breathing. So the adult hemoglobin you will find has a composition of 2 alpha, 2 beta, which is 97%. A very small component, which is also known as a minor hemoglobin, the alpha 2 and the delta 2, which gives rise to A2 concentration of less than 3.5%. And the fetal hemoglobin usually becomes less than 1% roughly by the age of 1 year. So these are just generalizations that you need to just kind of keep in mind. And basically what I'm just telling you is just revisions of what you may have just learned. So the human globin gene is one of the prototypic genes by which we try to understand the entire genetic code, because the entire genetic code was first deciphered in the globin chains. Why? Because it is one of the most easiest, proteins to be extracted. You just need a sample of blood from where you could get the globin chains. And also it's one of the most simple gene. So if you understand the basics of the genetics, which is there of the genes, you will find that this is the most simple one. If you understand this, then after that, when you start reading about different genes, you will find that each other genes are so complicated when it is compared with the human globin gene. So universally, though the locations of these globin chains are in different chromosomes, you will find that all the globin chains universally have 3 exons. So exon 1, exon 2, and exon 3. So between the exons, we know that there are the introns or the intervening sequences. So we have 2 intervening sequences, intervening sequence 1, which is much shorter than the very long intervening sequence 2. The other things to keep in mind is that the coding always starts with actually the ATG, which is coded by methionine. And before that, about 50 nucleotides before that, we call it the capsid, where from your mRNA, actually the mRNA starts getting translated. Now, there you can see that before that, there are certain sequences which are there, which are known as the sequences of the promoter region. So very typically, you would have remembered that, we say that we have the Tata box and things like that. So in the globin chains, there is no Tata box, but we have an ATA box. And this is just a nomenclature which is given. The promoter sequences will have a CA, triple C kind of a location. And these are sites which are very important, because you have the transcription factors which are going to give the green signal that this is the globin chain which needs to start synthesizing. So then you have the production of this mRNA, and after that you finally get a protein. After the splicing happens and the introns are excluded, you get a sequence like this, which is the 5' untranslated region starting from the cap site. And then you have the ATG, which translates to AUG, from where actually the first amino acid of the globin chain starts. And the color coded are such that the blue is the exon 1, which is getting translated. The exon 2 is in red, and the exon 3 is in green. And you have the stop codon, that is the UAA here, where actually the production of the coding will actually stop. And then you have a poly A signal here. At the end, you have a poly A tail. So this is the entire structure of the human mRNA, which is there, which gives rise to the protein. Finally, the translation happens, and the numbering actually of the protein is 146 proteins, because the first methionine actually is not numbered. Nowadays, we sometimes say the numbering starts from 1, and we say that the amino acid number is 147 in the beta globin chain. So this is just the very basics of how the globin chain is synthesized. Now, how does it translate to mutations? In the 6th codon, which is there in the beta globin, it is normally a GAG. Now, one nucleotide change can give rise to a mutation, as it happens in sickle cell anemia, where the center nucleotide, it becomes from A, it becomes a T. And you get just this one change gives rise to sickle kind of the sickling property to the hemoglobin molecule. Similarly, on the same codon, when you have the first nucleotide, which originally or normally is a G, if it becomes A, then it gives rise to a hemoglobin C. So you can see that when you have these changes which happen, so normally there is a production from GAG to glutamic acid, which is a negative charge. If it becomes a sickle cell hemoglobin, it becomes a valine, it becomes neutral. And if it becomes a hemoglobin C, then it becomes a lysine. So what does it translate to when we are looking at, as a pathologist in the lab? We find that these various amino acids, they have a particular mobility on the hemoglobin electrophoresis. So the glutamic acid will move fastest, and then you have the sickle cell, the sickle hemoglobin, which will migrate a little slower, and the slowest will be the hemoglobin C. So this is how the mutation will translate to an abnormal protein. Now, when do we say that the disorder is thalassemia? When there is a quantitative defect of the globin chain. So the nomenclature goes as, and the commonest hemoglobin disorder which is there is a thalassemia of the beta globin chain. So we call it a beta thalassemia, when the beta globin production is reduced. Similarly, the production of can be beta 0 when you get no production from a particular allele, or you can get some production which is known as beta plus, or you can get some more production which is known as beta plus plus. So in the nomenclature, you can see that the delta chain can also be affected. So if you get delta beta deletion, then we call it a delta beta type of thalassemia, and so on and so forth. Then hemoglobinopathies will be a qualitative change. So I said the hemoglobin changes, the amino acid changes, and like the sickle cell would be a abnormal hemoglobin or a qualitative change. We also have sometimes a situation where in adults you will find high fetal hemoglobin. Now we know that the high fetal hemoglobin is not really a abnormal hemoglobin, but the production or seeing it in more amounts than, say, the roughly 1% that we get in adults is known as an inappropriate expression. So all these disorders, the commonest being the thalassemias, the not so common hemoglobinopathies such as the sickle cell hemoglobinopathy, or the HBE kind of disorders with beta thalassemia, the combinations can give rise to patients requiring transfusions. The high fetal hemoglobin disorders are not very common. So I told you also when I was describing the structure of the hemoglobin molecule, that you can get abnormal proteins which can give rise to unstable hemoglobin disorders. You can get some gain of function variants which are known as polycythemic variants, and you can get some acquisition of unusual properties such as methemoglobinemia. Now, these three disorders, These are very sporadic hemoglobin variants, but they are inherited as autosomal dominant conditions. And the other common ones are inherited as autosomal recessive disorders. So this is a similar kind of whatever I have kind of told you till now, that if you get mutations, abnormal proteins, you can get loss of function, the commonest being thalassemias, or you can get unstable hemoglobins. And, some of the mutations can give rise to the gene dosage problems also, or an inappropriate expression, such as happens with hereditary persistence of fetal hemoglobin, also commonly known as HPFH, or the delta beta thalassemias, which are usually deletionals. Now, if we look at the abnormal hemoglobins which are there in India, majority of all over India, you get beta thalassemia, which is there. But hemoglobin E beta thalassemias more commonly are seen in the East. We also have a belt of sickle cell disorders. When you get either homozygous sickle or sickle with beta thalassemia, it's mainly towards the central India. Now, we also know that nowadays we have a lot of population migrations, and I, being a Bengali, I am in Punjab, so this kind of thing happens, and because of this, you get actually all the abnormal hemoglobins which can be seen in anywhere in India and anywhere now in the world as well. But within India, the kind of mutations which are there are more or less kind of uniform, and we'll talk about it a little later. We know that about 10,000 new cases of thalassemia major are added every year in India, and this is only a rough kind of population estimate which is there. We really don't know the real estimate which is there at the moment. Now, in beta thalassemia, I told you that you will have less amount of beta chains, and what happens is that the gamma chain production, which should normally stop when the child is born, continues to produce. So whatever little hemoglobin is formed is actually the fetal hemoglobin. And the problem is that the extra alpha chains, and I told you that alpha chains are anyway normally also in excess, so they will precipitate. And because of that, you get a lot of intramedullary death of the instability of these globin chains which are produced. In alpha chain disorders, usually they are deletional, as opposed to the mutations which are more common in the beta globin gene. And the nomenclature of the alpha gene disorders is such that normally we have 4 alpha genes which are functional. The silent alpha plus type of thalassemia is when you have 3 alpha globin genes which are functional. So basically we are saying that one is knocked down. We will call it a homozygous alpha plus when you get 2 functional genes which are there, but they are there in 2 different alleles. So we call it a homozygous alpha plus here, and this would be a heterozygous alpha 0 when normally like one of the alleles is like normal with 2 functional alpha genes, and the other 2 alleles are kind of alpha 0. So we'll say that this individual is alpha 0 compared to alpha plus when it is this kind of a situation. So this is also an alpha plus heterozygous, this is an alpha plus which is homozygous, and this is an alpha 0 which is heterozygous. So this really needs a little more kind of, going through the slide over and over again, or the text, to understand this, because it is just a kind of concept which is there important. Usually these kind of deletions are not very important, but HBH disease is a disease where the patient only has one functional alpha chain, and the patient presents usually like a thalassemia intermedia kind of a phenotype requiring transfusions. But we know that hemoglobin H disease is like a whole spectrum of disorders which are there, and patients can present at multiple ages, like various ages. Patient can be completely asymptomatic also, or sometimes during stress situations like say a pregnancy in a female, at that time the patient might require some transfusions. So there is a whole spectrum of HBH disease. Hemoglobin Barts is a condition which is usually lethal, and you have no alpha genes which are functional. Now, so this is kind of just the pathogenesis which is there, that the precipitation of the excess alpha chains gives rise to inclusion bodies or ineffective erythropoiesis and hemolysis. And You have bone disease, iron loading, and jaundice. And sometimes these patients can also have more numbers of episodes of infections. There are certain tertiary modifiers which are there, which can change the phenotype of the patient's kind of presentation. So if we look at the thalassemia major, these patients will have an early presentation.